So while they're getting set up, I'll just take a couple minutes to introduce David Granger. And I think one thing we can take away from Tanya's talk is how complicated the system is. And but with all the complexity, there's opportunity. And I know that, you know, we've already heard David Granger speak, so I'm not going to give a long introduction. But it's important to know that he's obviously a distinguished professor. He's been at that interface of materials and biology for a long time, but also at that interface of industry and academia. And also he's a very distinguished skier, so something that you might not know about him. I think we already know that you're a little bit curmudgeonly, and so...
Really?
So we are expecting you to take all that opportunity that Tanya highlighted and, you know, not depress us more, but lift our spirits and see the opportunities.
No one's ever accused me of lifting spirits. Wow. I'll try to wake up this audience. Thank you, Molly, and it's nice to be back. So I think Tanya did a great job of helping me get started. I'm going to focus on the clinical applications because I think this is a very nice analytical story for you. And so let's get started. First, we have a little quiz. Audience participation. What's the largest administration of nanomaterials to human tissues? What's the product? What's the application? Is it tumor delivery, like Tanya indicated? No, it's boring, right? It's cosmetics, lotions, and creams. Sunscreen, Shiseido is very expensive product in Japan, has gold and other metallic nanoparticles in it. All right, second question, what's the largest use of nanotechnology in nanomedicine? In other words, why do we use nanotechnology in nanomedicine? Was it to make nanoparticles to go into tumor formulations like Tanya described? What's your answer? In fact, it's mundane. It's drug solubility enhancement, and this has a long history. So if you think that nanomedicine's new, this is really a mundane story. There's a number of products whose sole purpose now, almost for 20 years, whose sole purpose is we just grind the drug to dust in order to increase its solubility. As you might know, also Tanya knows this very well, 70% of new drug candidates are insoluble in body juices. So one way to make it happen is to decrease the size. And that's a manifestation of what's called the Noyes-Whitney equation. If you change size here, you change surface area. And if you change surface area, then you can get more drug into solution in terms of what's called kinetic solubility. So in fact, most nanotechnology is used to grind particles to small sizes in order to get them to dissolve faster. All right, here are two classic anti-tumor drugs. Doxorubicin shown here, paclitaxel shown here. Both are sparingly soluble drugs. Both have very short circulating half-lives and relatively short elimination times. So these are the classic chemotherapy drugs that are used in very many solid tumors in almost all cancer clinics around the world. I'm not going to focus on this except to tell you that we can then use nanoparticles to change how these drugs are delivered. The problem with their current delivery of just sticking a needle in your arm or in your port and giving you chemotherapy is that most of the drug, as Tanya indicated, 1% of the dose of paclitaxel or Doxil will go to the tumor. The rest goes to where it's not supposed to go, to the lung, the heart, and the liver, and other places, and that's where side effects occur, and that's where toxicity occurs. So if 1% of the drug is going to where it's supposed to, it means that 99% of the drug is going to where it's not supposed to. So this is then the challenge of nanoparticles, is to change this, what we call biodistribution profile. Now, oncology drug sales a few years ago approached $100 billion, and this is the clinical gain that we're experiencing currently for that $100 billion of development. The median gain in progression-free survival for new cancer drugs approved between 2002 and 2014 was 2.1 months. Total overall survival for taking these drugs is 2.1 months. I would say, just to agree with Molly and be Mr. Happy for the moment, that that's a miserable outcome. I don't want to pay $60,000 for a years of chemotherapy to have a 2.1-month life extension. All right, so nanocarriers are then proposed to change how all of this operates. And the two poster children that we'll focus on here are Doxil, which is a liposomal form of doxorubicin, shown in purple, where that doxorubicin is insoluble, but it's sitting inside the basketball structure in an aqueous environment within the liposome. The second one is Abraxane, where the drug paclitaxel, which is not soluble, is physically encapsulated within an albumin microshell. There's no chemistry involved at all. We simply precipitate paclitaxel within an albumin microsphere, and in fact, it's well known that in the bloodstream, a lot of this paclitaxel diffuses out very rapidly, and it's well known that in the bloodstream, a lot of this doxorubicin diffuses out of the liposome very rapidly. So you could ask what the effect of the nanoparticle is. That's a different story. I will note that we have almost up to 50 nanomedicines now approved. They're either intravenous anti-cancer drugs or they're oral formulations of drugs that are meant to be solubilized by their very fine powder structure. Okay, the EPR effect, I don't have to cover this again. Tanya did a good job with that, but what I will focus on now is this idea that the randomized control trial is where these drugs and these formulations are tested in humans. Now, not everything is tested by RCTs. It should be, but in fact, they're very, very expensive and they're very tedious to design and operate. So only a very few things are tested by RCT testing. And when many things are tested around the world with the same drug and the same formulation, but one's done in Melbourne, Australia, and one's done in Tokyo, Japan, and one's done in Novosibirsk, then we can pool that randomized data and perform what's called a meta-analysis, in which we take the clinical results from all over the world and put them together and try to make sense to see if statistically these drugs are performing. All right, so let's look at some of that meta-analysis data as we now see it coming out for drugs like Doxil that have been around for 20 years. Meta-analysis starting to tell us something. I'll also then look at a meta-analysis for Abraxane, and I'll look at clinical results for a Korean drug called Genexol PM, a polymeric micelle formulation of paclitaxel. All right, so here's a meta-analysis of human nanomedicines and therapeutic use. It looks at 224 studies considered to date. 14 of those were randomized controlled trials, comprising almost 2,500 patients, and compared the clinical efficacy. It looked at overall response, that's called OR. It looked at overall survival, or OS, and it looked at progression-free survival, or PFS, in these systems. Those are the three classic clinical indicators that you can find all around the world that these studies then look at. All right, Doxil versus free doxorubicin drug. It's been done in mice for almost 30 years. There was an enormous amount of data that showed all of these benefits in mice with tumors when you graft mice ectopically, orthotopically, xenotopically, and so forth. All the different flavors of tumors. They even did also cardiotoxicity in dogs and rabbits, and they found substantial benefit in animals that compelled them then to move to humans. The meta-analysis showed higher accumulation in tumor than in any human data in the animals versus any human. All of a sudden, a huge difference between animals and human data. They decreased dose-limiting cardiotoxicity that's common for the free drug. That was one reason to formulate doxorubicin in a particle, was to get rid of the cardiotoxicity that's very commonly associated with the free drug. But we didn't find any dose-limiting toxicity in humans that was very comparable. Instead, we found then that we didn't have any mirroring of the preclinical data in showing increased efficacy versus free drug. In other words, humans showed none of the benefit of the animals. Most Doxil accumulates, as Tanya indicated, in places that are off-target. So you see this here. If you have a lung tumor, you get enormous amounts of radioactive drug accumulating in the spleen, and liver, the bone marrow, and also the lung. And a new toxicity showed up in humans, which never manifested itself ever in any animal trial, and that was the mucocutaneous toxicity, which is now well known for Doxil, that was never seen in any animal study. If we look at Abraxane versus free drug, paclitaxel, or what's called Taxol, again, preclinical animal studies showed enormous benefit Abraxane lowered the tumor volume compared to free drug at equal doses. Abraxane substantially improved tumor survival in many different types of ovarian cancer, breast cancer, uterine cancer, and other types of grafts in mouse models. But if we look at human data, the albumin carrier reduced the toxicity of the drug, so that was a benefit of the nanoparticle, was that by substituting this albumin over the traditional excipient used to solubilize Taxol, that the toxicities in humans were reduced. That was a thumbs up. But the higher, and therefore, because you reduced the toxicity, then you could produce a higher Abraxane dose. So you could increase the dose over the free drug substantially because the toxicity was reduced. So this was a benefit of the nanoparticle. However, if we now move from toxicity to efficacy, the efficacy in breast cancer patients was in fact only 5 weeks longer. 5 weeks longer for this expensive nanoparticle treatment compared to free drug. And Abraxane extended patient life by only 10 weeks as a second-line therapy if the primary therapy failed. So here's the meta-analysis data for Doxorubicin, comparing all of these studies, for cisplatin, which I don't have time to talk about, comparing all of these studies, and for paclitaxel and paclitaxel in this study here. And the meta-analysis here draws a simple line between the free drug or the nanoparticle drug. And if this line lies to the right substantially, then it favors the nanoparticle. And if it lies to the left, then it favors the free drug. And you can see here, given the error bars, that in cases of overall objective response, neither Doxil, nor cisplatin, nor paclitaxel had any distinguishing meta-analysis benefit for overall response in human patients. If we look at the overall survival, here's the meta-analysis data, favors the nanoparticle, favors the free drug, and you can see this lies right on the line. There is no difference between free drug and nanoparticle in overall survival in all of these nanoparticle drug classes. And if we look at progression-free survival in humans treated with these, we can see again, everything lies on the line. There is no difference in all three of the clinical indicators between liposomal doxorubicin, Abraxane nanoparticles versus free drug, in addition to cisplatin, which I didn't get a chance to talk about. So what's the cost difference between free doxorubicin chemotherapy and Doxil chemotherapy? What's the multiplier? What do you think it is? It's not 1,000, but it's between 10 and 100, depending on which clinic you go to. So you're paying 100-fold more for no benefit. Is that bright and cheery for you, Amali? Okay. All right. So now we look at Japan. Here's Nanocarrier, a company that I know quite well in Japan, and they invented this polymeric micelle formulation, conceptually shown here, carrying paclitaxel. And they called it NK105, and they conducted then an RCT, very expensive multi-center trial. Phase 3, that's the epoch, highest level of efficacy testing in humans. And their preclinical data in animals showed again really impressive data for tumor models in mice, enhanced circulation time, enhanced kinetic profiles, solubilizing the drug without this toxic excipient, preventing human sensitivity reactions. And this was all published. So they run the clinical trial, and the indication is for metastatic recurrent breast cancer. And on July 6th, last year, 2016, they issued this statement: Nanocarrier's position, and announced that the results of the phase 3 study for NK105 in patients with recurrent breast cancer could not achieve the primary endpoint. Nanocarrier, basically they terminated the phase 3 trial. So what does that indicate? If I had moved through, this probably cost $100 million to date. If I had moved that company through to phase 3 and then had to issue this press release, that means that somehow the results in humans were very bad. All right, but it goes further. Here's a company founded by a prominent member of the National Academy in my country. Founded a company in Boston. They raised $80 million in private money, and they went public in 2014. They created this polymer drug conjugate that comprises a cyclodextrin and the cancer drug here, shown in this motif, where the cancer drug is linked with a cleavable linker, camptothecin, to cyclodextrin and PEG copolymer. And because of the hydrophobicity of this drug, it forms these nanoparticles. And there is an enormous amount of literature published on this chemistry, on this preclinical data shown here, where almost every type of tumor in every type of mouse has been tested and shown repeatedly that if you treat, in all cases, a single treatment cycle of 3 weekly doses resulted in significant anti-tumor effect. Complete tumor regression was observed in all animals bearing, and so you can see tumor regression here with the treatment, tumor regression here with the treatment, tumor regression here with the treatment. And here's the Kaplan-Meier curves showing almost 100% survival of these mice treated with these fatal tumors, treated with this polymeric nanoparticle construct. Interesting. March 2017, get this press release that says that Cerulean's polymer nanoparticle failed 2 randomized controlled phase 2 trials. NASDAQ Cerulean is selling off all of their drug assets, and they will become overnight a new company that sells contraceptive vaginal rings. Wow. I mean, if I was an investor in that company, I'd be calling my lawyer really quick, because this is an about face in which they sold, after raising over $150 million from investors, they sold off their assets. 1.5 million went to this company and 6 million went to Novartis. If I invested $150 million in that, it was in phase 2 trials, and now I have a vaginal contraceptive ring company, I'd be very unhappy. Does that make you happy, Molly? Okay, so let's talk about this Korean company, Samyang Biopharma. Samyang has already on the market selling Genexol, which is a classic paclitaxel chemotherapy using this toxic excipient to solubilize it. So there's the toxic excipient, this Solian oleate, and paclitaxel here. That's the conventional infused formulation. So they went and made a nanoparticle formulation that is poly-DL lactide, and it solubilizes the paclitaxel inside the core of the micelle, and they call it Genexol PM because it's a polymeric micelle. Okay, great. Here's their human data. Overall survival. Red is the conventional paclitaxel formulation. Blue is their polymeric micelle formulation. Overall human survival. Progression-free survival. Red is the free drug. Blue is the nanoparticle. And then down here is the table where the polymeric micelle adverse events, neutropenia and hypersensitivity, which are 2 very common reactions to paclitaxel infusion. And what's interesting is that there is no difference in overall survival. There is no difference in progression-free survival. There is, in fact, an enhancement of grade 3 neutropenia, an enhancement of grade 2 hypersensitivity compared to the free drug. So not only are the outcomes of efficacy the same, but the adverse events are worse for the nanoparticle formulation. So here's the only case of a targeted nanoparticle, and it's Merrimack Pharmaceuticals in Boston, in which they took liposomal formulation of doxorubicin, shown in red here with the liposome in purple, and put then the Herceptin anti-breast tumor antibody on the surface. Logical concept, clever concept, been reported in the literature many, many times. 2017, Merrimack Pharmaceuticals halts phase 2 study of their HER2 antibody-targeted liposomal doxorubicin. There were no safety concerns, so toxicity was not an issue, but there was no benefit over the comparator, which was Doxil, untargeted, and Herceptin antibody, no liposome. So they couldn't show any benefit against the 2 products that were already out there. Stopped. All right, so this prompted then this reaction from Chemical Engineering News in my country. Does nanomedicine have a delivery problem? And this was an editorial that was written. And it was then questioning our peer-to-peer credibility. Do we really report the right data? Do we design the right experiments? When 100% of 6 mice live in a cohort, does that justify moving to human clinical trial? I think not. But then this article went further to talk about business credibility. If our science can't be backed up by our human clinical data, and yet we create businesses from such concepts, then we ruin the connection between science and business, which is translation, which is a topic that we're going to discuss later today and later this week. So this idea of translation from science, how we represent ourselves, the credibility of what we do, and how it actually impacts the human as opposed to publishing a Nature paper or publishing a Journal of Controlled Release paper, is another topic for another day, but I think it's worthy of some thought. All right, but there's an upside. So this company, Celator, New Jersey, unknown company, found this magic formula of making a liposome where if you put 5 red dots and 1 blue dot together inside a liposome, that you can get, in fact, a very effective anti-cancer medicine. So, in fact, it's a first version of a co-encapsulated drug where 5 cytarabines and 1 Doxorubicin, which is a Doxorubicin knockoff, 5-to-1 ratio, produces a synergistic anti-tumor effect. Now, who would have thought, right? But this company found this out, and they got fast-track status both in the United States and Europe to treat AML, which, because of its desperate therapeutic status, deserves some attention. It was granted fast-track designation, and it got approval, and instantly, as soon as Celator got approval on the fast track for this drug, it was bought by Jazz Pharmaceuticals for $1.5 billion. This is like a company with like 13 people in it. Yeah. So anyway, it's an amazing story of success, but it's a non-intuitive success where somebody found this magic ratio, and now it's in humans to treat a very desperate disease. So that's an upside. Molly, you should be smiling. So, current nanomedicines, the last slide. Right now, they show little improvement in efficacy. I can't find any efficacy data out there in human trials of any statistical significance to refute this claim. But what we have learned so far is that we can modify dosing. We can give patients more drug without toxicity because the nanoparticle changes the route of distribution, and it changes how particles move in the human body, and it reduces toxicities that are well known for the free drugs, so it allows us to give more drug using this biodistribution. And so Kinab Park last year, the editor of Journal of Controlled Release, he basically wrote this up with me and has this table summarizing these problems of all these different nanoparticle drugs done by smart people in academic labs and translated poorly, though, to humans, and how they terminated their phase 1, 2, and 3 trials. So, lies, damned lies, and statistics. I think it's easy to show in your Ph.D. work, and in my lab, and anyone's lab, that you can push nanoparticles across various barriers. I think you can push them across cell barriers. I think you can push them across rat intestines. I think you can push them through nasal mucosa. You can push them through the blood-brain barrier. Yeah, I think it's statistically possible that you can do that. It's possible, but that's not the translational game here. The translational game is showing that it's therapeutically relevant, that it's clinically reliable and therapeutically important. So if you're playing this statistics game, you can publish the papers all you want, and you can put them into mice models and get the data that you want for your PhD thesis. But you have to do more than that to get clinically reliable data. Statistics doesn't work for humans. So I guess that's the message, is that we have to think about what we're doing, and this was a discussion at my table at lunchtime, to think about what we're doing and why we're doing it. And if we're doing it just to publish papers, then yeah, we can focus on statistical likelihoods. But if we're doing it to fix patients' lives and improve the degree of medical care, then I think we have to think about those elements. And right now, that system seems to be broken in many, many ways, and I think we need to fix it. So with that, I'll stop and take questions with Tanya. She can come up here, and happy to move on. Thanks. See, I am Mr. Happy.
David, thank you very much. Lived up to our expectations, although you did leave us with a glimmer of hope. And we have time for questions.
Viola.
Thank you, Dave, for another really important talk. My question in the moment is that the community addresses now quite often what goes wrong in the design of nanoparticles, what did we not understand in terms of transport of nanoparticles into dense cancer tissue. But the question that is not asked very often is whether the community uses the right animal models. So I think on the animal side, there's an intrinsic difficulty. The intrinsic difficulty is we always use young mice. We induce the tumor. It grows really fast. It typically grows within a week or two to measurable size. Then we apply the treatment. Whereas what we try to treat later on are older patients with really aged matrix, with slowly grown tumors that morphologically look completely different from mouse tumors. And we know that upon aging, cross-linking of matrix changes, the entire matrix composition changes, and all these other factors. So if we continue to spend all the billions on mice studies, we probably will never make progress in terms of finding a good treatment for human diseases that occur at advanced age.
Excellent. I couldn't have said it better. I had 4 slides this morning because of time. I just skipped through 4 animal study slides, which say essentially exactly that. That, in fact, yeah, I don't have to repeat it. Your point's very well taken. So my only response is that we've learned a lot from mice, and it's time to move on. The poor assessment I have is that everybody I know who has proposed a large animal model for tumors, because large animals don't grow tumors spontaneously, except for dogs, and dogs are an ethical problem to do research on, and I agree with this. But now we have the CRISPR rabbit, we have the CRISPR pig coming down line, and so there are ways now to create humanized animals that will grow tumors that are human-sized at human rates. But if you've ever done a large animal study, you know how expensive just small cohorts are. So we can get rid of mouse studies, and it's going to flush, 1/3 of these students in this room, it's going to flush part of their Ph.D. thesis. We have to think about what we're going to do with that. But then we have to add a 0 to every research grant in terms of the budget and say, okay, I'm going to do 30 CRISPR rabbits instead of, 7,000 mice, and see what happens, because we don't know. But I must say that every time I've been in study section where a tumor experiment has been proposed on a CRISPR rabbit or a CRISPR pig, it's been adamantly declined. So we'll see how long this takes to get hold. I don't know how else to do this, but I think that mice are no longer a relevant model for therapeutic designs. I think there's a lot of utility for mice in lots of other ways, but in the way you talked about it, I think you're right on.
I think we should probably, as a community, make more efforts to maybe work with veterinarians. And I assume that many of us having really old animals, and if they go into stages of major suffering, that we would be glad to test the drug to see whether their status can be improved.
Yeah, the problem is
at least those are really slowly grown tumors to make a first step. But then certainly the issue about reproducible and is always there. And
maybe we'll thanks so much. Maybe we'll move to the next question.
Maybe a little bit. I think the picture you described was a little bit too dark, because none of the drugs actually made the situation worse. So none of the formulations made it worse. So I think it's actually a major achievement that you package and you don't make it any worse. Maybe you should start to allow human trials sooner, because you don't make it worse. I think that's one. And the second comment is that, yes, we could stop doing mice, but first we should probably study what is the difference between the study that works and the study that doesn't work, because the studies that don't work in mice don't get published.
That's right. So negative results are another big problem, which we've talked about a lot, in terms of not knowing what you should know because it's not publishable. But there should be a database somewhere where negative results are shown so that we don't repeat ourselves, and we can mine that negative data to see why it was negative.
And it really can be a very simple reason why all these trials don't improve, and it works in mice. Just we don't know what it is.
Yeah, very well put too. Those are two, for those in the audience, those are two very common responses to this talk and two very important, totally valid points.
But maybe on the hopeful side, Tanya, you wanted to maybe add something?
These were more in vivo points, so I mean that's fine.
Use the mic.
Fully addressed, yeah.
So, actually, if you don't mind, I'll take the chair's
Is
prerogative here.
your mic on?
Is this mic on?
There we go.
Okay. Yeah, so I'll take the chair's prerogative. Another hypothesis here would be not that nanotechnology has failed, but that the drugs have failed, that you're actually trying to pursue nanotechnology with drug molecules that are intrinsically flawed. And so what we see at the end of the day is results that perhaps are not that terribly surprising. And kind of as a counter to the example you're giving, I'll think about immunotherapy, which is also based almost entirely on rodent models. It's been wildly successful in terms of really extending lives, curing patients. So using the same models in many cases, obviously there's many things that haven't translated, but some things have translated that are tremendously important. And so maybe it's really the biology and the drug molecules we're choosing to pursue as much of the problem as the technology itself.
I think Tanya can comment on the drugs. The only quick thing I'll add is that, you know, solid tumors really have not had impressive immunotherapy to date. Liquid tumors, yeah, but solid tumors have enormous side effects and not a lot of great efficacy data.
Well, actually, I mean, if you look at the PD-1, CTLA-4, broadly efficacious against solid tumors.
Not all solid tumors.
Not all, but
Lung
many,
carcinoma.
to many solid tumors. I mean, they're approved based on their data with melanoma, for example,
Yeah.
where you take a condition that is lethal to patients, uniformly lethal, and you get 25 to 40% of patients going on to live long term. That's a tremendous impact in a solid tumor.
Right, I was thinking more of the CAR T-cell therapies too, but okay.
Yeah, I think CAR T cells is certainly in the
Challenging
blood cancer
zone,
area.
yeah. Do you want
Yeah, but I would also agree, so that immunotherapy that offers also many opportunities nowadays. Also to use the carriers we've been using and evaluating before. I think that knowledge is not lost. It's not, I mean, we gained a lot of experience, as Dave has also very nicely already shown, so on circulation times, on stability and so on. In immunotherapy, you also, there's a big impact on sizes of the nanocarriers, so whether basically they are then uptaken by macrophages and so on. So you can, I think you can use those concepts and basically see how well it works later on for delivery in immunotherapy. And there are very promising studies out. I mean, we need to see how they develop. Yeah, there will also be pitfalls. But so I think so. It's not that negative that we didn't learn anything. So we have I think we have a solid basis now also on what kind of materials basically have a toxic response. So where do we see toxicity towards so in the blood and towards blood cells and basically enrichment in other organs and so on. And you can use this knowledge maybe later on for different approaches like immunotherapy and maybe also for others. And also with respect to drug design, I would also say biologicals are emerging there. So there are, for example, toxins, protein toxins, bacterial toxins that are super efficient, so just a couple of molecules that need to enter a cell in order to inhibit Rho and interact with intracellular pathways in a way small molecules cannot do. So I think there are also opportunities of new concepts emerging. Yeah, so I think one should still be, in that sense, positive that we learned something also from these negative studies that has been shown, but we also need to take that as a basis of knowledge and also be honest. I absolutely agree. We need to be honest and say, so what has failed so far? Because otherwise, people would also lose trust in chemists designing carrier systems and so on. I think we learned a lot and we should use this knowledge now also for other directions and other developments and hopefully perform better there.
Hi. Do you think that this massive poor efficacy could be mainly due to the fact that really little is known about the variability in the human cases, especially in a field like cancer? Really little is known on a biological characterization of the variability patient to patient. And then if after this will be possible to do and characterize, do you think that everything else could be adapted accordingly. Even this small animal model, if you actually understand all the biological behind the variability, can you adapt even the small animals for that, and then treat and then test your treatments?
I think Tanya very effectively in her talk addressed the fact that tumor heterogeneity, there's no such disease as cancer, right? And even in lung cancer, it's extremely variable. And even within a lung cancer within a patient, one single patient over space and time, cancer is not cancer. There's different expression of receptors, there's different necrotic regions, different vascular zones, and that changes in a patient all the time. So the heterogeneity, I think, cannot be overemphasized. You're very correct there. How that heterogeneity is expressed in syngeneic mice, super dosed with potent malignant cells, in their hindquarter. I don't know how to characterize the heterogeneity, but certainly all of those issues are a big problem with where we are with animal models versus how to translate and understand and translate that to humans, as Tanya showed.
Maybe we could have one last question from the monitor here. There's a couple that crossed over, so I hope you apologize. I didn't ask your question, but they crossed over with the others. But maybe you could each give a short answer to something I think is very pertinent to your talks today, which is, what is your advice to PhD students working in this space today?
You go first.
Maybe it's not so long ago that I've been a PhD student in this area. So yeah, it's difficult to answer. I think if you focus on translation in your PhD, I think that's very challenging. But I think there's a lot to discover also on the molecular level. So I'm a chemist, I can only give a chemistry approach. So basically how molecules can be designed with respect to shape, with respect to distances of ligands at the surface to interact efficiently with receptors to understand basically how to enter cells and how to distribute there. I think there's a lot that can be done, and I've seen nice concepts, for example, where people use origami, so DNA as a tool to position ligands, also diverse ligands in a very distinct fashion, and then investigate interactions with receptors, investigate probably uptake, and to enhance our understanding, for example, of ligand receptor interactions at the macromolecular scale. So I would personally I think you always need, as a PhD student, you are probably part of a team, but you would need to solve your own scientific problems. So I think I think one also needs to break it down to a problem that is solvable for a PhD student. And I think so if one investigates how molecules, macromolecules can be designed, interact with cells, how they can traffic inside the cell. We didn't discuss that at all, but which molecule can you control by macromolecular design, whether a molecule ends up in the mitochondrium or goes somewhere else, and then investigate how the cell interacts. I would find that stimulating. So I think there are concepts in that direction. I would, for PhD thesis, I would find basically still quite attractive.
Maybe a shorter answer from you, David.
Yeah, if that's short, I get a long time.
Yeah,
okay. So I think it depends on the question you're asking. And I do think that mechanistic questions for PhD students are much better than efficacy questions. The efficacy question cannot be answered in a cell culture dish. Targeting cannot be answered in a cell culture dish. Disease treatment cannot be measured in a mouse, relevant disease treatment to humans. So if those are off the table, and you can convince your PI or your advisor that those are off the table, then there is a lot of other questions that can be asked using genetic models, mutant models, different receptor models, and biophysics, biochemistry, to get a very meaningful PhD thesis in cell culture and in a mouse. But efficacy is probably not a good question. So.
Okay, so I have the opportunity to close this session. I think really what we've seen in this section is that there are a lot of challenges, but there's also a lot of opportunity and a lot of opportunity for creativity. And so with those challenges of whether you're trying to get into the cell or you're trying to do an animal study, you don't want to do the same thing that everybody's done, but come up with your own take in your own creative way. So with that, thank you very much to the speakers, and we have a break.